So good morning, afternoon or evening, depending on where in the world you are joining us from today. My name is Jeremy Dumsday. I'm the Director of the Sales for Twist Bioscience in the CSIRO region. Firstly, I would like to extend a warm welcome to you all and thank you for joining us for this exciting webinar today. Synthetic biology continues to move forward at rapid speed, enabled by the iterative cycle of design, build, test, learn for DNA and strain engineering. At the BioFoundry in the Commonwealth Scientific and Industrial Research Organisation, or CSIRO, scientists have used automation and modular workflows to enable a high throughput build stage, as well as associated design, test and learn capabilities to be offered to their customers. In this Twist hosted webinar, we'll be joined by two leading scientists from the CSIRO to discuss these cutting edge capabilities. Before introducing our guest speakers, just a few housekeeping items. All lines will be muted during the webinar. We will answer all questions at the end of the presentation. If you have any questions, please submit in the Q&A box rather than the chat box. And lastly, following the presentation, if you could please take 1 minute for a brief survey, we would really appreciate the feedback. It gives me great pleasure to introduce today's guest speakers. Maciej Holowko, or Magic as we like to call him, is a synthetic biology stack developer at the CSIRO. Maciej obtained his Masters of Engineering degree from Warsaw University of Technology and his PhD from the Nanyang Technology University in Singapore, where he developed the first whole-cell biosensor for in vitro seek and kill of Vibrio cholera. In his current role as the synthetic biology stack developer, his work focuses on the development of high-throughput synthetic biology technologies, including the emerging BioFoundry based in Brisbane, Australia. Our second guest speaker, Janet Reid, is the BioFoundry Facility Manager at the CSIRO. Janet is an experienced molecular and biologist with a microbiology degree from the University of Queensland, a graduate diploma in biotechnology and a PhD in prostate cancer-associated proteases from the Queensland Institute, University of Technology. Prior to the BioFoundry, Janet worked for the Community for Open Antimicrobial Drug Discovery, a not-for-profit initiative at the Institute for Molecular Bioscience at the University of Queensland, Australia. In this group, she developed high-throughput human cell screening protocols, assessing effects of antimicrobial and anti-inflammatory compounds. Without further ado, I welcome Maciej and Janet to present Full Steam Ahead, Taking Synthetic Biology to the High-Throughput Era.
Hi, welcome everyone. Thanks, Jeremy, for the kind introductions. As Jeremy mentioned, myself and Janet, we are part of the CSIRO, the Commonwealth Scientific and Industrial Research Organisation. We are Australia's national science agency, and as one, CSIRO acknowledges our first nationals people, the traditional owners of the land, sea, and waters of the areas that we live and work on across Australia. We acknowledge their continuing connection to their culture, and we pay our respects to their elders, past and present. My name is Maciej, as Jeremy mentioned, but please feel free to call me Magic. That's usually easier and easier to remember. So yeah, let's go on with the presentation. So a brief word about CSIRO itself. As I mentioned, we are Australia's national science agency. We are around for almost 100 years now, and we are a very big organization. We have over 5,500 dedicated people all across Australia. And for you connecting with us today from the US, the easiest comparison for you would be that CSIRO is kind of similar to the National Labs system, where we are directly funded by the government, but we also work with industry a lot. So it's estimated that we are delivering $4.5 billion of value to the Australian economy through our science and technology. And as I've mentioned, we are sites all over Australia and in all states and territories. So Australia is also the innovation, CSIRO is Australia's innovation catalyst. We have quite a few of patent families. We have licenses. We have also a portfolio of startup and more established companies. And we work with over 2,000 different partners locally, domestically, and overseas. And we also have quite big education wing where we work with over 150,000 school students. So let's go and talk about synthetic biology, because I believe many of you know what synthetic biology is, but for those of you who don't, synthetic biology is extension of the genetic engineering, as it was called previously, where we take living organism and we try to make them to make something useful for us. So the basic is the basis is we take sugars, light, and biowaste, things that are available in bulk, especially in Australia when it comes to sugars and light. For example, in Queensland, there is the biggest production of cane sugar in the world. And so the point is, synthetic biology, we take the organism and we make them to produce pharmaceuticals or nutraceuticals, fibers, textiles, agriculture, chemicals, flavors, fragrances, personal care, colorants. And all those stuff, of course, are chemicals. So the gist of it is rewire whatever is in the cells, in the original cells, be it bacteria, yeast, or some more advanced cells, and rewire in a way, so we change, for example, the metabolism, or we add genes, we remove genes to make them produce something that is actually useful to us, so we can extract it and use in commercial way. It was recognized by CSIRO almost 10 years ago now that synthetic biology is a very important and emerging science. And CSIRO decided to create a program called the Synthetic Biology Future Science Platform, where the goal was to bring synthetic biology into Australia. The program itself is based at CSIRO, but we work a lot with Australian universities all around Australia again. There were a number of projects and fellows that were brought into Australia under this program, where they were based in all the universities around the country. And the idea was for them to just start the synthetic biology programs in those universities. Now, after a few years of the program, we were very successful in bringing this science, synthetic biology, into Australia. And so those are projects. There were some projects in CSIRO itself as well delivered. And one of those big projects that were delivered within CSIRO itself was the BioFoundry, about which I will be talking in a moment. So what actually is the BioFoundry? So BioFoundry is an automated high-throughput laboratory which uses standardized protocols and workflows to deliver synthetic biology. So currently, you probably know that synthetic biology is delivered in a more artisanal, bespoke way, where you have a scientist that goes into the lab, works at the workbench, creates all the genetic constructs that they need and an organism and tests them. The problem is the throughput, of course, and the time that it takes. So you have your scientists they are very valuable, of course, and they use most of their time to just work in the lab, which is not very efficient. And automation is a solution to that. The problem is, of course, that before you can automate something as advanced as genetic engineering or synthetic biology, there is a lot of work that has to be done to make it work. And that's what biofoundries are all about. They are trying to create those environments, the automated environments for scientists to work in, so they don't have to focus on doing the lab work. The lab work will be handled by the robots, and they can focus on the important stuff. So there are quite a few biofoundries around the world currently, both commercial and academic, and they are being prototyped. So they are prototyped because where we currently are trying experimental fabrication methods. So what we are all trying to achieve is to check how to bring synthetic biology into this high throughput era. So our protocols and ways of standardization are being developed, but at this point, we are already much faster and more efficient than a standard lab. So we believe that we are still in this experimental phase, but we are on cusp of going into this mass production phase, which is where we want to get eventually, right? So this is very similar to a lot of other industries around the world, where first people have to figure out how to do stuff and in high volume, and then there's explosion of production. And finally, you reach the maturation where you produce as much as can be really produced. And as I said, BioFoundry is just on cusp of going into this mass production phase. So one word about the Global BioFoundries Alliance. So it's a group of academic biofoundries from all around the world that includes Northern America, Europe, Asia, and Australia. We actually have 2 biofoundries in Australia. One is in Macquarie University in Sydney, and the other one is in Brisbane, Queensland. So the main interest of this alliance is to jointly develop hardware and software and business models and policies and different protocols. So the development of biofoundries around the world is kind of standardized as well. So this is where we go into our biofoundry. So our biofoundry is based in Ecosciences Precinct, Brisbane, Australia. So Brisbane is the capital of the state of Queensland. It was initially a CSIRO and the University of Queensland joint facility. And the main goal of the facility is to bring the state-of-the-art synthetic biology platform technology to Australia. And with this, I'll pass to Janet.
Thanks for that, Maciej. Now, just check that everyone can hear me okay. Just a nod. Yep, great. Thanks, Maciej. So the BioFoundry essentially is a bioengineering facility, service facility, and we take your designs and adapt them for our high throughput processes and machinery. We can assemble DNA, transform organisms, and provide some phenotyping and data analysis. And we provide these services to the synthetic biology community. So in this section of the presentation, I'll discuss these aspects in more detail, and we can consider the benefits of our services to the types of research that you do. So many of you, as Jeremy mentioned already, many of you were familiar with the design, build, test, learn cycle. And it's the principle by which we conduct our work in the BioFoundry. It's an iterative process in which designs are based on the learning from previous design, build, and test cycles. And in practice, we found we are in the BioFoundry, In practice in the BioFoundry, we're layering a number of aspects, beginning with the materials that we choose, the DNA in the cells and the molecular biology kits that either designed for or adapted to high throughput processes. And we test various kits and cell lines to find optimal solutions. Layer on that the hardware in the BioFoundry. We acquire high throughput automated or semi-automated instruments. And the types of instruments we acquire are driven by market research. We've talked to members of the synthetic biology community and other biofoundries and developed a consensus list for us to pursue in our purchases. Layer on that the software necessary for the high throughput design and instrument maintenance and management and data handling. And finally, on top, the point of it all, the application and our service delivery is to customers that can be from industry or academia or anywhere in the team in between. So let's pause for a moment and consider this question. What are the roadblocks to you using high throughput processes to execute your engineering projects? Is it equipment access? Is it price, expertise, intellectual property? And by this we mean, do you have concerns about allowing access to your IP by others? And E, other. Many of you are recording your responses at the moment. If you find you'd like to expand on your response or add anything, please feel free to use the chat or Q&A buttons and add anything you'd like on there. Looks like we have another 20 seconds to go, and we're trending towards equipment access and expertise as the leaders on the board here. Equipment access it is. Luckily, you've come to this presentation today then, hey? Okay, now let's move on to the next slide. Now, let's delve down a little bit into what we can offer in our service pipeline and about some of the complementary services that can be found in CSIRO that can also contribute to the pipeline. Sorry, for some reason my slides flipped twice. Excuse me, I think there's some lag. There we go. So first off, you can come to us and we can have a chat about what you need, the consultation process. We can talk about what processes we have or what we can develop for you to get your desired outcomes. We also aim to offer automation training, but that part of our dream is on hold due to the instability in the COVID situation. And we are looking to do virtual education programs through CSIRO. They already run these through high schools at the moment, so this format could work for us. Next, we can provide feedback on cloning strategies in the design phase. And we can also do batch design of DNA constructs and organisms using our software. These strategies feed into the build phase, which I'll go into in more detail in the following slides. And for the testing phase, we have some basic phenotyping, and also CSIRO has breadth and depth of testing capabilities that can be exploited to add to this pipeline. For example, next door to our lab here in Ecosciences, there's a metabolomics facility that can be accessed. We also, inside CSIRO Victoria, have bioreactor facilities. Now, the linchpin to this cycle, the DBLT, is the learn. And Maciej's been working closely with Data 61 on this, which is another arm of CSIRO, to develop a component of this pipeline. So let's go into it further. I'll give you a breakdown on some of the software and instruments that we use. So the DNA synthesis is the first, DNA design and synthesis is the first stage of the process. But the backbone for the Foundry is the Tecellogen software. We use the design software as well as build modules, which in the design software, we can perform routine cloning through to advanced scale combinatorial and hierarchical designs. And the output of this module is a list of DNA parts and primers required for the design. And depending on the design strategy, these lists can be forwarded to your favorite synthesis company or PCR generated. The other Tecellogen module that we use, the build module, is essentially a LIMS system, laboratory information management, massive inventory and workflow management system. In it, we can enter all our laboratory equipment, track the storage of items, track change of states of samples, use of primers, dilutions of aliquots, transformation of DNA into organisms and recovery back out again. Workflows can be planned in this software and executed, generating plate maps and integrating our automation. The data from phenotype and genotype studies can also be uploaded into this software. It's a very powerful tool and designed specifically for the synthetic biology engineering workflows. And the great thing about Tecellogen is they're open to feedback and incorporate changes and updates to their software based on the user experiences. They've got a further 2 modules in development which will address test and learn. They call it test and evolve, because they've recognized the need for big data management and processing in these 2 parts of the cycle. So we're looking forward to seeing what those modules offer. So delving further into the build flow, so we use a number of different types of assembly methods to produce our DNA constructs, including Gibson, Golden Gate, and most recently we've been testing Nuge. It's a new method which has come from Twist Biosciences, developed by Rebecca Nugent, and it's somewhat similar to Gibson. All our work is in 96 well format and the reduced volumes for assembly. So we use the Echo liquid handling instrument down here for our array of fragments, DNA fragments in PCR plates. It's capable of dispensing in nanoliter volumes. So typically I'd use a 1 microliter reaction volume for assemblies. After thermocycling for assembly, we then use the Felix liquid handler and the cyclone for organism transformation processes. We can also use them for DNA extraction processes. As you can see from the image up here, we can use this instrument to array microliter dots of transformed organisms on selection agar. Once these colonies have grown, we can then use our colony picking robot, the Pixel, and it's a fantastic instrument, this one. It has multiple wavelength light sources and filters, and it has AI built into the software. So it allows a certain level of trainability, and this combination allows for selection of colonies based on a range of characteristics, including the size, the shape, color, and fluorescence. So, continuing the workflow and introducing a couple more of our instruments. In the test phase, we screen colonies based on genotype and phenotype. For example, we may use colony PCR to quickly identify if DNA of interest has been introduced into our strains. And again, this is in the 96 well format. We have the ZAG DNA analyzer that we can use that has capillary gel electrophoresis to separate the DNA based on molecular weight and quantify it relative to a DNA ladder. And as you can see from the image of the instrument, you may be able to see these drawers. So we can put plates in each of those drawers so you can run plates back to back. It's essentially a very high throughput instrument. So in addition to this basic genotyping in this rapid screening method, we can also extract the plasmid or genomic DNA for sequencing by an external provider. On the phenotyping side, we've got a multi-mode plate reader, the Citation, and for colorimetric and flowmetric analysis of reporter molecules that you may have engineered into your strains, or we can use a Cytoflex flow cytometer for population studies. And we'll be adding a fax machine to this cohort of instruments in the coming months. And this upcoming purchase of a fax machine is based on feedback from the community about what type of services they would like in the BioFoundry. As mentioned, a machine learning pipeline is in development by Maciej in conjunction with Data61. And to add to this, one of the promises of the test and evolve packages of Teselagen is the ability to feed in the data and exploit the machine learning algorithms that they are developing. So let's go to another poll question. If you're thinking about taking up a service like ours, what's the most important thing to you? Is it throughput, turnaround time, price, expertise, scientific support, service offering? And by this we mean services available in the pipeline or other. And again, if you've got anything to contribute to further, please put it into the chat or Q&A. And looks like we have a lead for expertise and scientific support at the moment. It's edging out price. Oh, looking good. Yep, there we go. Expertise and scientific support. That's great to hear. Thank you for the responses on that. So let's move on to the advantages of the BioFoundry. So one of the main advantages, of course, is the high throughput that the BioFoundry affords. But the instrumentation and the data capture capabilities can lend themselves to exploration of hypotheses that would have been far more challenging to examine if you're using less automated or low throughput methods. So essentially, we can all boil this all down to the advantages down to the reduction in time to identify candidate strains. In industry, this can equate to a reduction in time to market. So now let's just have a look at this and think about what you might like to do with our facility. So we're open to introducing new methods. For instance, a project that's coming up will be introducing the U-loop cloning system. And what type of organisms do you want to work with? And think about what project you've had in the back of your mind, but the scale of it has thwarted your execution. You know, you can come and talk to us and let us see if we can help. Now, this is the final poll question. How likely are you to use a BioFoundry, the BioFoundry, our BioFoundry for your project? Would you like to sign up today? 3 months? Or do you need some more lead time, like 6, 12 months? Or maybe not that likely? Maybe E, did you just crash this Zoom meeting and you don't actually know what's going on? Oh, it looks like somewhat likely is leading the pack here. Looking good, almost 50%. This is great. Keep the responses coming. Fantastic. Heading out to 6 to 12 months, somewhat likely. A little bit of lead time. You need to do some planning to get into there. And that's probably very good. If you have something on the go ready to introduce to us, please come and talk to us. But if it's something that you need a little bit more lead time, please talk to us again and we can start planning out. So for my last couple of slides, I'm just going to introduce the rest of our team. So in the lab with us, we've got Hugh Heyman-Zumpe. He dabbles in a bit of everything in the lab, but he has skills in yeast engineering that he's been putting to good use on projects that we've been running. Our direction is guided by Colin Scott and Alison Rice, and Claudia Vickers is stepping back from the role in the Foundry to continue her work guiding the CSIRO SynBio FSP to a greater extent. But these are all enthusiastic advocates for the BioFoundry's purpose. And our purpose is to accelerate development in bioengineering and reducing time to market for products. So I'd like to thank you for attending the seminar today. Please keep questions coming. To wrap up, a very quick thank you to CSIRO and UQ. And also thank you to our hosts today, Twist Biosciences, for allowing us to present the BioFoundry to you in this forum. The team at Twist have provided us with enthusiastic support throughout this process. And over to you, Jeremy. Thank you.
Thank you, Maciej and Janet, for an outstanding and informative presentation. We will now open up to some question time, which we have about 15 minutes allocated for. If you have any questions, can you please type them in the Q&A box on the webinar? We have a few questions in there already, and I will put some of those forward to Janet and Maciej now. Please feel free to enter more as we go on. The first question comes from Nigel. What is CSIRO's annual budget? That might be a tough one.
Yeah, I can answer this one, I think. Currently, it's just shy of 1 billion Australian dollars from the government, but that's not the whole budget. We get around 40% extra on top of that from our collaborations with industry. Collaborations, big contracts. So yeah, around 1.5 billion total, I believe.
Okay, thank you. And from Aziz, how much can BioFoundry cost? What sort of resources do you need to bring this to a community?
Well, it can cost anything really, from very cheap to very expensive. So I can shamelessly advertise that we'll be soon publishing a paper about how to approach starting BioFoundry. It will be published at the Oxford University Press in Bio. So keep on lookout on that. But I would say you would need a couple of millions to really get everything going, and there's a significant time where you won't be producing much. You'll be just trying to figure things out, and we need personal stuff for that, and machines, of course, have to be there. So yeah, a couple of millions, unless you want to do it very bespoke. It depends also on the scale, right? Because we are gearing towards working with industry and academic clients. So our throughput has to be quite high, but I believe you could set it for much lower if, for example, you just want to work with your academic lab or within your department, or it really depends. But yeah.
Okay. A question from Joanne. Do you have any intellectual property restrictions for the people that use your systems? And perhaps you can explain, if you do, how you get around that or how you work with that.
Janet?
You're on mute, Janet.
Sorry, I was reading questions in the Q&A. What was the question again, please?
Do you have intellectual property restrictions for the people that use your systems, your service?
So restrictions. I'm not sure what you mean by restrictions, but because we're a service facility, we're going for the approach of your intellectual property is your intellectual property. If that's the gist of the question. Of course, there will be some situations where we might enter a collaboration with particular groups, and then the negotiation may pan out in a different way. But strictly as a service facility, IP is yours, except for the IP to actually run the BioFoundry processes. That's our IP.
Yeah. A technical question: which 96-well DNA isolation kits do you find work best, especially for E. coli?
So far, we've been using the Invitrogen kits, and we're going to start using, so that was their column-based kit. Can't remember the name of it quite offhand, but we're going to use their MagBead kit next as well. And we've got another MagBead kit coming from the Zymo research as well, which we've had some good results from some of the other Zymo products. So we're hoping that will be good.
All right, Janet. Which step in the cloning workflow benefits the most from automation, in your opinion?
Well, I guess all of them really, because it's pretty much the same process, but multiplied, right? Because, well, yes, all of them really, because you just take your normal reaction, your transformation reaction, so you have to prepare your DNA, you have to clean it up and then put together with cells and transform, incubate and everything. So really, it's parallel process. It doesn't speed it up because you have to do things like, run your assembly reaction. You have to do your outgrowth after transformation and everything. So that you cannot cut down this time. But what happens is, because you're not doing 5 transformations, you're doing 100 or 200, right? So that's where the advantage is.
Maybe the lab staff would say the colony picking aspect, because they don't have to do it.
I was going to say colony picking, and actually the echo for arraying DNA for assembly is also pretty awesome. But at those volumes, brilliant.
All right, thank you. So the next question from an anonymous is: what is your current design build cycle time before reaching test, assuming you can offer just high-throughput DNA construction?
Construction plus organism transformation. If it's just standard, it can be done in a week, depending on the organism, of course. If it's something with a slow growth cycle, then that's longer, but mostly it's about a week to get to the test stage.
Okay. Joanne's got another question. Hi there, the BioFoundry sounds great. Some questions. What organisms can we use? Bacteria, yeast, tissue culture, i.e. plants or animal cells?
So currently we work only with bacteria and yeast and diatoms. So those are the three classes. We cannot process mammalian cells or any other higher organisms yet, maybe in the future. So those are our limitations. So currently it's mostly for microorganisms.
Okay. Another anonymous, could I please ask the false positive rate for DNA assemblies?
That really depends on the assembly methods. We found that there's variation depending on the strategies that people have employed. So yeah, that's really a strategy question. Yeah.
Do you work with industry? Companies work with foundries for DNA construct synthesis? From Michael Hudson.
Industry? Yes. Sorry,
yes.
Yes.
Okay, we've got a lot of questions coming in. I don't think we're going to get through all of them with the we've got left. We'll move on to Karen Weinberg's question. Will there be more flexibility in allowing biological entities such as bacteriophages to be engineered in the BioFoundry? Are there considerations given to potential contamination by other organisms?
Hi, Karen. Hi. At this stage, no, there isn't any plan to introduce the phage or any other organism like that into the BioFoundry. Yes, I think that question is still, it hasn't been resolved.
It might be depending on the project again. What type of budget ballpark price should a project need to be able to utilise the BioFoundry? Yes,
definitely project dependent, because it depends how much scientific consultation is involved, because we have all of the CSIRO standing behind us. So if you need help with aspect A, B, and C with molecular engineering or whatever you need, we can factor that into our contracts. Also the throughput, the volume. Of course, the bigger the volume, the lower the price per sample to a point. And also, if a project would be of scientific interest to CSIRO, there are other ways to negotiate collaborations. So it really depends on the size of the project, but in general, the bigger the better. And again, it depends also how much of scientific expertise do you require. But I would really encourage the person asking just to send us an email, and we can come up with a rough estimate based on your requirements.
Okay. A question from Michael. Got a question on how can CSIRO services be extended to other countries, for instance, Africa?
Good question. I think we are currently open for overseas clients. So I don't see this as a problem. So if you would like to use our facility, again, just let us know. There are, of course, biosecurity and biosafety questions to be answered for that. But yeah, definitely open to discuss this.
We would consider export and import permit issues as well, so depending on whether you're planning to send us material to work on or whether we have everything synthesized here on as well would make a difference to accessibility of the foundry for external clients.
You mentioned that you are outsourcing your sequencing. Can that be integrated or automated?
Definitely.
Yes. We've been thinking about it. It's something that we've considered. We're trying not to do everything here as well. So we're trying to think about what parts of the pipeline we definitely need to keep as core facility services here and what can be farmed out to other facilities or groups.
Yes, we've recently acquired the Oxford Nanopore system. We don't know how much of a role it will be playing in the BioFoundry yet, but yeah, definitely sequencing, as Janet mentioned, is going to be part of our workflow.
Okay. Great question from Philip. How have you approached predictable scaling of bioprocesses utilizing engineered strains? What is the largest scale you have of your study?
Scale as in a volume of, sorry again?
I would say in terms of size.
Of the volume of the culture, or how many samples do we process? I'll answer both probably. So we can easily process a couple hundred samples per week, most probably, when it comes to production of the genetically modified organism. And then it depends what do you really need. If you need bioreactor checks and that kind of scale up, we have a dedicated facility in Victoria, in Melbourne, to do that. So we would just send your samples there and they would be processed there for the scale up. And we are also hoping to acquire a mini bioreactor system for our own facility. So we can do a small scale up tests and then send your samples on to Victoria.
Okay. We have time for 2 more questions, so apologies for any of those that haven't been answered live. We will endeavour to get to those by responding in text. Another question from Nigel. What size of constructs are you able to build? Can you reach the size of secondary metabolite biosynthetic clusters of 50 to 100 kb?
We haven't worked with such a big project yet, but definitely I don't see that as a big problem with the use of technology. That shouldn't really be any barrier. So definitely open to such a project. We are currently doing some genome engineering as well. So definitely doable for us. Just a question of what do you exactly need and how we can do that.
Okay. And I promise I'm not showing any favoritism here. It was next in the queue. Can you explain how the BioFoundry and Twist are working together?
Yes. So as Janet mentioned as well, we work with a number of synthetic DNA manufacturers. And if Twist products are the best for what you need, then we will just work with Twist to get you the best product for you.
Okay.
As mentioned as well, in this, we were testing the new product as well. We're one of the beta testers and very happy to have been asked to do that as well.
Okay. Right, we're just about out of time. Once again, I would like to thank our guest speakers, Janet and Maciej, and normally would invite you all to do so with a round of applause, but good old COVID right now is preventing us from doing that. A quick reminder that following this webinar, you will be redirected to a brief survey. If you could please take 1 minute, that would be greatly appreciated. And finally, thank you all for attending today's webinar. I hope you found it insightful and informative. Have a great day, evening, or night ahead, and thank you.
Thank you, everyone.
Thank you very much.